Scientists who follow Darwin continue to make the same error. The problem with this underemphasis on the environment is that it led to an overemphasis on nature in the form of genetic determinism. The belief that genes control biology. This belief has not only led to a misallocation of research dollars, as I will argue in a later chapter, but more importantly, it has changed the way we think about our lives. When you're convinced that genes control your life and you know that you had no say in which genes you were saddled with at conception, you have a good excuse to consider yourself a victim of heredity. Don't blame me for my work habits. It's not my fault. I've been procrastinating on my deadline. It's genetic. Since the dawn of the age of genetics, we have been programmed to accept that we are subservient to the power of our genes. The world is filled with people who live in constant fear that, on some unsuspecting day, their genes are going to turn on them. Consider the masses of people who think that they're ticking time bombs. They wait for cancer to explode in their lives as it exploded in the life of their mother or brother or sister or aunt or uncle. Millions of others attribute their failing health not to a combination of mental, physical, emotional, and spiritual causes, but simply to the inadequacies of their body's biochemical mechanics. Are your kids unruly? Increasingly, the first choice is to medicate these children to correct their chemical imbalances, rather than fully grappling with what is going on in their bodies, minds, and spirits. Of course, there is no doubt that some diseases like Huntington's chorea, beta thalassemia, and cystic fibrosis can be blamed entirely on one faulty gene. But single gene disorders affect less than 2% of the population. The vast majority of people come into this world with genes that should enable them to live a happy and healthy life. The diseases that are today's scourges, diabetes, heart disease, and cancer, short-circuit a happy and healthy life. These diseases, however, are not the result of a single gene, but of complex interactions among multiple genes and environmental factors. What about all those headlines trumpeting the discovery of a gene for everything from depression to schizophrenia? Read those articles closely, and you'll see that behind the breathless headline is a more sobering truth. Scientists have linked lots of genes to lots of different diseases and traits, but scientists have rarely found the one gene that causes a trait or a disease. The confusion occurs when the media repeatedly distort the meaning of two words, correlation and causation. It's one thing to be linked to a disease, it's quite another to cause a disease, which implies a directing, controlling action. If I show you my keys and say that a particular key controls my car, you at first might think that makes sense because you need that key to turn on the ignition. But does the key actually control the car? If it did, you couldn't leave the key in the car alone because it might just borrow your car for a joyride when you are not paying attention. In truth, the key is correlated with the control of the car. The person who turns the key actually controls the car. Specific genes are correlated with an organism's behavior and characteristics, but these genes are not activated until something triggers them. What activates genes? The answer was elegantly spelled out in 1990 in a paper entitled Metaphors and the Role of Genes and Development by H.F. Nieschut. Nieschut presents evidence that the notion that genes control biology has been so frequently repeated for such a long period of time that scientists have forgotten it is a hypothesis, not a truth. In reality, the idea that genes control biology is a supposition, which has never been proven and, in fact, has been undermined by the latest scientific research. Genetic control, argues Nieschut, has become a metaphor in our society. We want to believe that genetic engineers are the new medical magicians who can cure diseases and while they're at it, create more Einsteins and Mozarts as well. But metaphor does not equate with scientific truth. Nieschut summarizes the truth. When a gene product is needed, a signal from its environment, not an emergent property of the gene itself, activates expression of that gene. In other words, when it comes to genetic control, it's the environment, stupid. Protein, the stuff of life. It is easy to understand how genetic control becomes a metaphor as scientists with ever greater excitement zeroed in on the mechanisms of DNA. Organic chemists discovered that cells are made up of 4 types of very large molecules: polysaccharides, complex sugars, lipids, fats, nucleic acids, DNA, RNA, and proteins. Though the cell requires each of the 4 molecular types, proteins are the most important single component for living organisms. Our cells are, in the main, an assembly of protein building blocks. So one way of looking at our trillion-celled bodies is that they are protein machines, although, as you know, I think we are more than machines. It sounds simple, but it isn't. For one thing, it takes over 100,000 different types of proteins to run our bodies. Let's take a closer look at how our cells' 100,000 plus proteins are assembled. Each protein is a linear string of linked amino acid molecules, comparable to a child's pop bead necklace as illustrated below. Each bead represents one of the 20 amino acid molecules used by cells. Though I like the pop bead analogy because everyone is familiar with it, it is not an exact one because each amino acid has a slightly different shape. So, to be completely accurate, you should think of a pop bead necklace that got mangled a bit in the factory. And to be even more accurate, you should know that amino acid necklace, which forms the backbone of the cell's proteins, is far more malleable than a pop bead necklace, which falls apart when you bend it too much. The structure and behavior of the linked amino acids in the protein backbones better resemble that of a snake's backbone, as shown below. The spine of a snake, made up of a large number of linked subunits, the vertebra, is capable of coiling the snake into a wide variety of shapes, ranging from a straight rod to a knotted ball. The flexible links, peptide bonds, between amino acids is a protein backbone and enable each protein to adopt a variety of shapes. Through the rotation and flexion of the amino acid vertebrae, protein molecules resemble nanosnakes in their ability to writhe and squirm. There are 2 primary factors that determine the contour of a protein's backbone and therefore its shape. One factor is the physical pattern defined by the sequence of differently shaped amino acids comprising the pop bead-like backbone. Unlike uniform-shaped pop beads, each of the 20 amino acids comprising protein backbones has a unique shape or conformation. Consider the differences between the character of a backbone made from identically shaped pop beads and one assembled from the differently shaped pipe fitting illustrated above. The second factor concerns the interaction of electromagnetic charges among the linked amino acids. Most amino acids have positive or negative charges, which act like magnets. Like charges causes the molecules to repel one another, opposite charges cause molecules to attract one another. As shown on the following page, a protein's flexible backbone spontaneously folds into a preferred shape when an amino acid subunit rotate and flex their bonds to balance the forces generated by their positive and negative charges. The protein backbones shown in A and B have the exact same amino acid pipe fitting sequence above, but reveal radically different conformations. Variations in the backbone's shape result from different rotations at the junctions between adjacent fittings, like pipe fittings. The protein's differently shaped amino acids also rotate around their junctions, or peptide bonds, allowing the backbone to wiggle like a snake. Protein shapeshift, though, they will generally prefer 2 or 3 specific conformations. Which of the two conformations, A or B, would our hypothetical protein prefer? The answer is related to the fact that the two terminal amino acids, or pipe fittings, have regions of negative charges. Since like charges repel each other, the farther apart they are, the more stable the conformation. Conformation A would be preferred because the negative charges are farther apart than they are in B. The backbones of some protein molecules are so long that they require the assistance of special helper proteins called chaperones to aid in the folding process. Improperly folded proteins, like people with spinal defects, are unable to function optimally. Such aberrant proteins are marked for destruction by the cell. Their backbone amino acids are disassembled and recycled in the synthesis of new proteins. How proteins create life. Living organisms are distinguished from non-living entities by the fact that they move. They are animated. The energy driving their movements is harnessed to do the work that characterizes living systems, such as respiration, digestion, and muscle contraction. To understand the nature of life, one must first understand how protein machines are empowered to move. The final shape, or conformation, or the technical term used by biologists, of a protein molecule reflects a balanced state among its electromagnetic charges. However, if the protein's positive and negative charges are altered, the protein backbone will dynamically twist and adjust itself to accommodate the new distribution of charges. The distribution of electromagnetic charge within a protein can be selectively altered by a number of processes, including the binding of other molecules or chemical groups such as hormones. The enzymatic removal or addition of charged ions or interference from electromagnetic fields, such as those emanating from our cell phones. Okay. Figure A shows the preferred conformation of our hypothetical protein backbone. The repelling forces between the two negatively charged terminal amino acids, see the arrows, causes the backbone to extend so that the negative amino acids are as far apart as possible. Figure B shows a close-up of amino acid. Figure B shows a close-up of an end amino acid. A signal, in this case, a molecule with a very positive electric charge, the white sphere, is attracted to and binds with the negative site on the protein's terminal amino acid. In our particular scenario, the signal is more positive in charge than the amino acid is negative in charge. After the single signal couples with the protein, there is now an excessive positive charge at this end of the backbone. Since positive and negative charges attract one another, the backbone's amino acids will rotate around their bonds so that the positive and negative terminals will come closer together. Figure C shows the protein changing from conformation A to conformation B. Changing conformations generates movement, and the movement is harnessed to do work, providing for such functions as digestion, respiration, and muscle contraction. When the signal detaches, the protein returns to its preferred extended conformation. This is how signal-generated protein movements provide for life. The shape-shifting proteins exemplify an even more impressive engineering feat because their precise three-dimensional shapes also give them the ability to link up with other proteins. When a protein encounters a molecule that is a physical and an energetic complement, the two bind together like human-made products with interlocking gears, say an egg beater for an old-fashioned watch. Say an egg beater or an old-fashioned watch. Examine and following two illustrations. The first shows 5 uniquely shaped proteins, examples of the molecular gears found in cells. These organic gears have softer edges than machine shop manufactured gears, but you can see that their precise three-dimensional shapes would enable them to securely engage with other complementary proteins. So the picture is a protein menagerie. Illustrated above are 5 different examples of protein molecules. Each protein possesses a precise three-dimensional conformation that is the same for each copy of itself in every cell. A is an enzyme that digests hydrogen atoms. B, woven filament of collagen protein. C, channel, a membrane-bound protein with hollow core pore. D, protein subunit of capsule that encloses a virus. And E, DNA synthesizing enzyme with attached helical DNA molecule. In the second illustration, page 29, I chose a wind-up watch to represent the workings of the cell. The first picture shows a metal machine revealing the gears, springs, jewels, and case of the watch model. When gear A turns, it causes gear B to turn. When B moves, it causes gear C to turn, etc. In the next image, I overlay the human-made machine gears with softer edge organic proteins, magnified millions of a time in proportion to the watch, so that it becomes visually conceivable that proteins could be like the watch's mechanisms. In this metal protein machine, one can imagine protein A rotating and causing protein B to revolve, which in turn causes protein C to move. Once you see that possibility, you can look to the third figure in which the human-made parts are removed. Voila! We are left with a protein machine, one of the thousands of similar protein assemblies that collectively comprise the cell. Cytoplasmic proteins that cooperate in creating specific physiological functions are grouped into specific assemblies known as pathways. These assemblies are identified by functions such as respiration pathways, digestion pathways, muscle contraction pathways, and the infamous energy-generating Krebs cycle, the bane of many a science student who has to memorize every one of its protein components and complex chemical reactions. Can you imagine how exciting cell biologists were when they figured out how the protein assembly machine works? Cells exploit the movements of these protein assembly machines to empower specific metabolic and behavioral functions. The constant shape-shifting movements of proteins, which can occur thousands of times in a single second, are the movements that propel life. The primacy of DNA. You'll notice that in the above section, I didn't discuss DNA at all. That's because it is the changing of the protein's electromagnetic charges that is responsible for the behavior-generating movement, not DNA. How did we get to the widespread and often cited notion that genes control biology? In The Origin of Species, Darwin suggested that hereditary factors were passed from one generation to generation, controlling the traits of the offspring. Darwin's influence was so great that scientists myopically focused on identifying that hereditary material which they thought controlled life. In 1910, intensive microscopic analyses revealed that the hereditary information passed on generation after generation was contained in chromosomes, thread-like structures that become visible in the cell just before it divides into two daughter cells. Chromosomes are incorporated into the daughter cell's largest organelle, the nucleus. When scientists isolated the nucleus, they dissected the chromosomes and found that the hereditary elements were essentially comprised of only two kinds of molecules, protein and DNA. Somehow the protein machinery of life was entangled in the structure and function of these chromosome molecules. The understanding of the chromosome's functions was further refined in 1944 when scientists determined that it was DNA that actually contained hereditary information. The experiments that singled out DNA were elegant. The scientists isolated pure DNA from one species of bacteria, let's call it species A, and added the pure DNA to cultures containing only species B bacteria. Within a short time, species B bacteria began to show hereditary traits that were formerly seen in species A. Once it was known that you needed nothing other than DNA to pass on traits, the DNA molecule became a scientific superstar. It was now left to Watson and Crick to unravel the structure and function of that superstar molecule. DNA molecules are long and thread-like. They are made from 4 nitrogen-containing chemicals called bases: adenine, thymine, cysteine, and guanine, or A, T, C, and G. Watson and Crick's discovery of DNA structure led to the fact that the sequence of the A, T, C, and G bases in DNA spells out the sequence of amino acids along a protein's backbone. Those long strings of DNA molecules can be subdivided into single genes, segments that provide the blueprint for specific proteins. The code for recreating the protein machinery of the cell had been cracked. Watson and Crick also explained why DNA is the perfect hereditary molecule. Each DNA strand is normally intertwined with a second strand of DNA, a loosely wrapped configuration known as the double helix. The genius of the system is that the sequences of DNA bases on both strands are mirror images of each other. When the two strands of DNA unwind, each single strand contains the information to make an exact complementary copy of itself. So, through a process of separating the strands of a double helix, DNA molecules become self-replicating. This observation led to the assumption that DNA controlled its own replication. It was its own boss. The suggestion that DNA controlled its own replication and served as the blueprint for the body's proteins led Francis Crick to create biology's central dogma, the belief that DNA rules. The dogma is so fundamental to modern biology, it is essentially written in stone, the equivalent of science's 10 commandments. The dogma, also referred to the primacy of DNA, is a fixture of every scientific text. In the dogma's scheme of how life unfolds, DNA perches loftily on top, followed by RNA. RNA is the short-lived Xerox copy of DNA. As such, it is the physical template encoding the amino acid sequence that makes up a protein's backbone. The primacy of DNA diagram provides the logic for the age of genetic determinism. Because the character of a living organism is defined by the nature of its proteins, and its proteins are encoded in the DNA, then by logic, DNA would represent the first cause, or primary determinant of an organism's traits. The Human Genome Project. After DNA achieved superstar status, the remaining challenge was to create a catalog of all the genetic stars in the human formation. Enter the Human Genome Project. A global scientific effort began in the late 1980s to create a catalog of all the genes present in humans. From the outset, the Human Genome Project was a massively ambitious one. Conventional thought held that the body needed one gene to provide the blueprint for each of the 100,000 plus different proteins that make up our bodies. Add to that at least 20,000 regulatory genes, which orchestrate the activity of the protein encoding genes. Scientists concluded that the human genome would contain a minimum of 120,000 genes located within the 23 pairs of human chromosomes. But that wasn't the whole story. A cosmic joke was unfolding. One of those jokes was periodically unsettle scientists convinced that they have discovered the secrets of the universe. Consider the impact of Nicholas Copernicus' discovery, published in 1543, that the Earth was not the center of the universe, as was thought by the scientists, theologians of the day. The fact that the Earth actually revolved around the Sun and that the Sun itself was not the center of the universe undermined the teachings of the Church. Copernicus' paradigm-busting discoveries launched the modern scientific revolution by challenging the presumed infallibility of the Church. Science eventually displaced the Church as Western civilization's source of wisdom for understanding the mysteries of the universe. Geneticists experienced a comparable shock when, contrary to their expectations, over 120,000 genes, they found that the entire human genome consists of approximately 25,000 genes. More than 80% of the presumed and required DNA does not exist. The missing genes are proving to be more troublesome than the missing 18 minutes of the Nixon tapes. The one gene, one protein concept was a fundamental tenet of genetic determinism. Now that the Human Genome Project has toppled the one gene for one protein concept, our current theories of how life works have to be scrapped. No longer is it possible to believe that genetic engineers can, with relative ease, fix all our biological dilemmas. There are simply not enough genes to account for the complexity of human life or of human disease. Picture. The central dogma. The dogma, also referred to as the primacy of DNA, defines the flow of information in biological organisms. As indicated by the arrows, the flow is only in one direction, from DNA to RNA and then to protein. The DNA represents the cell's long-term memory, passed from generation to generation. RNA, an unstable copy of the DNA molecule, is the active memory that is used by the cell as a physical template in synthesizing proteins. Proteins are the molecular building blocks that provide for the cell's structure and behavior. DNA is implicated as the source that controls the character of the cell's proteins, hence the concept of DNA's primacy, that literally means first cause. I may sound like Chicken Little shouting at the genetics sky, that the genetic sky is falling. However, you don't have to take my word for it. Chicken Big is saying the same thing. In a commentary on the surprising results of the Human Genome Project, David Baltimore, one of the world's preeminent geneticists and a Nobel Prize winner, addressed the issue of human complexity. But unless the human genome contains a lot of genes that are opaque to our computers, it is clear that we do not gain our undoubted complexities over worms and plants by using more genes. Understanding what does give us our complexity, our enormous behavioral repertoire, ability to produce conscious action, remarkable physical coordination, precisely tuned alterations in response to external variations of our environments, learning, memory, need I go on, remains a challenge for the future. As Baltimore states, the results of the Human Genome Project force us to consider other ideas about how life is controlled. Understanding what gives us our complexity remains a challenge for the future. The sky is falling. In addition, the results of the Human Genome Project are forcing us to reconsider our genetic relationship with other organisms in the biosphere. We can no longer use genes to explain why humans are at the top of the evolutionary ladder. It turns out There is not much difference in the total number of genes found in humans and those found in primitive organisms. Let's take a look at 3 of the most studied animal models in genetic research. A microscopic nematode roundworm known as, oh goodness, I can't even say it, or the fruit fly, and a laboratory mouse. The primitive roundworm serves as a perfect model for studying the role of genes in development and behavior. This rapidly growing and reproducing organism has a precisely patterned body comprising of exactly 969 cells and a simple brain of about 302 cells. Nonetheless, it has a unique repertoire of behaviors, and most importantly, it is amenable to genetic experimentation. The Anaerobidus genome consists of approximately 24,000 genes. The human body comprises of over 50 trillion cells and contains only 1,500 more genes than the lowly spineless 1,000-celled microscopic worm. The fruit fly, another favored research subject, has 15,000 genes. So, the profoundly more complicated fruit fly has 9,000 fewer genes than the more primitive roundworm. And when it comes to the question of mice and men, we might have to think more highly of them or less of ourselves. The results of parallel genome projects reveal that humans and rodents have roughly the same number of genes. Cell Biology 101. In retrospect, scientists should have known that genes couldn't provide the control of our lives. By definition, the brain is the organ responsible for controlling and coordinating the physiology and behavior of an organism. But is the nucleus truly the cell's brain? If our assumption that the nucleus and its DNA-containing material is the brain of the cell, then removing the cell's nucleus, a procedure called enucleation, should result in the immediate death of the cell. And now for the big experiment. Maestro, a drum roll if you please. The scientist drags our unwilling cell into the microscopic operation arena and straps it down. Using a micromanipulator, the scientist guides a needle-like micropipette into position above the cell. With a deft thrust of the manipulator, our investigator plunges the pipette deep into the cell's cytoplasmic interior. By applying a little suction, the nucleus is drawn up into the pipette, and the pipette is withdrawn from the cell. Below the nucleus-engorged pipette lies our sacrificial cell, its brain torn out. But wait, it's still moving. My God, the cell is still alive. The wound has closed, and like a recovering surgical patient, the cell begins to slowly stagger about. Soon the cell is back on its feet. Okay, it's pseudopods, fleeing the microscope's field with the hope that it will never see a doctor again. Following enucleation, many cells can survive for up to 2 or more months without genes. Viable enucleated cells do not lie about like brain-dead lumps of cytoplasm on life support systems. These cells actively ingest and metabolize food, maintain coordinated operation of their physiological systems, respiration, digestion, excretion, motility, etc., retain an ability to communicate with other cells, and are able to engage in appropriate responses to growth and protection requiring environmental stimuli. Unsurprisingly, enucleation is not without side effects. Without their genes, cells are not able to divide, nor are they able to reproduce any protein parts they lose through the normal wear and tear of the cytoplasm. The inability to replace defective cytoplasmic proteins contributes to mechanical dysfunctions that ultimately result in the death of the cell. Our experiment was designed to test the idea that the nucleus is the brain of the cell. If the cell had died immediately following enucleation, the observations would have at least supported that belief. However, the results are unambiguous. enucleated cells still exhibit complex, coordinated, life-sustaining behaviors, which imply that the cell's brain is still intact and functioning. The fact that enucleated cells retain their biological functions in the absence of genes is by no means a new discovery. Over 100 years ago, classic embryologists routinely removed the nuclei from dividing egg cells and showed that a single enucleated egg cell was able to develop as far as a blastula, an embryonic stage consisting of 40 or more cells. Today, enucleated cells are used for industrial purposes as living feeder layers in cell cultures designed for virus vaccine production. If the nucleus and its genes are not the cell's brain, then what exactly is DNA's contribution to cellular life? Enucleated cells die, not because they have lost their brain, but because they have lost their reproductive capabilities. Without the ability to reproduce their parts, enucleated cells cannot replace failed protein building blocks, nor replicate themselves. So, the nucleus is not the brain of the cell. The nucleus is the cell's gonad. Confusing the gonad with the brain is an understandable error, because science has always been, and still is, a patriarchal endeavor. Males have often been accused of thinking with their gonads, so it's not entirely surprising that science has inadvertently confused the nucleus with the cell's brain. Epigenetics, the new science of self-empowerment. Genes as destiny theorists have obviously ignored 100-year-old science about enucleated cells, but they cannot ignore new research that undermines their belief in genetic determinism. While the Human Genome Project was making headlines, a group of scientists were inaugurating a new revolutionary field in biology called epigenetics. The science of epigenetics, which literally means control above genetics, profoundly changes our understanding of how life is controlled. In the last decade, epigenetic research has established that DNA blueprints passed down through genes are not set in concrete at birth. Genes are not destiny. Environmental influences, including nutrition, stress, and emotions, can modify those genes without changing their basic blueprint. And those modifications, epigeneticists have discovered, can be passed on to future generations as surely as DNA blueprints are passed on via the double helix. There is no doubt that epigenetic discoveries have lagged behind genetic discoveries. Since the late 1940s, biologists have been isolating DNA from the cell's nucleus in order to study genetic mechanisms. In the process, they extract the nucleus from the cell, break open its enveloping membrane, and remove the chromosomal contents, half of which is made up of DNA and half of which is made up of regulatory proteins. In their zeal to study DNA, most scientists threw away the proteins, which we now know is the equivalent of throwing the baby out with the bathwater. Epigeneticists are bringing back the baby by studying the chromosomes proteins, and those proteins are turning out to play as crucial a role in heredity as DNA. In the chromosome, the DNA forms the core, and the proteins cover the DNA like a sleeve. When the genes are covered, their information cannot be read. Imagine your bare arm as a piece of DNA representing the gene that codes for blue eyes. In the nucleus, this stretch of DNA is covered by bound regulatory proteins, which cover your blue eye gene like a shirt sleeve, making it impossible to read. How do you get that sleeve off? You need an environmental signal to spur the sleeve protein to change shape, i.e. detach from the DNA's double helix, allowing the gene to be read. Once the DNA is uncovered, the cell makes a copy of the exposed gene. As a result, the activity of the gene is controlled by the presence or absence of the ensleeving proteins, which are in turn controlled by the environmental signals. The story of epigenetic control is a story of how environmental signals control the activity of genes. it is now clear that the primacy of DNA chart described earlier is outmoded. The revised scheme of information flow should now be called the primacy of the environment. The new, more sophisticated flow of information in biology starts with the environmental signal that goes to a regulatory protein, and only then does DNA, RNA, and the end result a protein. The science of epigenetics has also made it clear that there are two mechanisms by which organisms pass on hereditary information. Those two mechanisms provide a way for scientists to study both the contribution of nature, or genes, and the contribution of nurture, or epigenetic mechanisms, in human behavior. If you only focus on the blueprints, as scientists have been doing for decades, the influence of the environment is impossible to fathom. Let's present an analogy, which hopefully will make the relationship between epigenetic and genetic mechanisms clearer. Are you old enough to remember the days when television programming stopped after midnight? After the normal programming signed off, a test pattern would appear on the screen. Most test patterns look like a dartboard with a bullseye in the middle, similar to the one pictured on the following page. Think of the pattern of the test screen as the pattern encoded by a given gene, say one for brown eyes. The dials and switches of the TV fine-tune the test screen by allowing you to turn it on and off and modulate a number of characteristics, including color, hue, brightness, contrast, and vertical and horizontal holds. By adjusting the dials, you can alter the appearance of the pattern on the screen, while not actually changing the original broadcast pattern. This is precisely the role of regulatory proteins. Studies of protein synthesis reveal that epigenetic dials can create 2,000 or more variations of proteins from the same gene blueprint. Here's the picture. Describing the picture. In this epigenetic analogy, the test pattern on the screen represents the protein backbone pattern encoded by a gene, while the TV's controls can change the appearance of the pattern, picture B and C. They do not change the original pattern of the broadcast, i.e. the gene. Epigenetic control modifies the readout of a gene without changing the DNA code itself. Next section. Parental life experiences shape their children's genetic character. We now know that the environmentally influenced fine-tuning described above can be passed from generation to generation. A landmark Duke University study published in August 1st, 2003 issue of Molecular and Cellular Biology found that an enriched environment can even override genetic mutations in mice. In the study, scientists looked at the effect of dietary supplements on pregnant mice with the abnormal agouti gene. Agouti mice have yellow coats and are extremely obese, which predisposes them to cardiovascular disease, diabetes, and cancer. In the experiment, one group of yellow obese agouti mothers received methyl group-rich supplements available in health food stores: folic acid, vitamin B12, betaine, and choline. Methyl-rich supplements were chosen because a number of studies have shown that the methyl chemical group is involved with epigenetic modifications. When methyl groups attach to a gene's DNA, it changes the binding characteristics of regulatory chromosomal proteins. If the proteins bind too tightly to the gene, the protein sleeve cannot be removed and the gene cannot be read. Methylating DNA can silence or modify gene activity. This time, the headlines, "Diet Trumps Genes," were accurate. The mothers who got the methyl group-rich supplements produced standard lean brown mice, even though their offspring had the same agouti genes as their mothers. The agouti mothers who didn't get the supplements produced yellow pups, which ate much more than the brown pups. The yellow pups wound up weighing almost twice as much as their lean pseudo-agouti counterparts. There's a picture. Agouti sisters, 1-year-old female genetically identical agouti mice. Maternal methyl donor supplementation shifts coat color of the offspring from yellow to brown and reduces the incidence of obesity, diabetes, and cancer. The university's photo on the previous page is striking. Though the two mice are genetically identical, they are radically different in appearance. One mouse is lean and brown, and the other mouse is obese and yellow. What you can't see in the picture is that the obese mouse is diabetic, while its genetically identical counterpart is healthy. Other studies have found epigenetic mechanisms to be a factor in a variety of diseases, including cancer, cardiovascular disease, and diabetes. In fact, only 5% of cancer and cardiovascular patients can attribute their disease to heredity. While the media made a big hoopla over the discovery of the BRCA1 and the BRCA2 breast cancer genes, they failed to emphasize that 95% of breast cancers are not due to inherited genes. The malignancies in a significant number of cancer patients are derived from environmentally induced epigenetic alterations and not defective genes. The epigenetic evidence has become so compelling that some brave scientists are even invoking the L word for Jean-Baptiste de Lamarck, the much-scorned evolutionist who believed that traits acquired as a result of environmental influence could be passed on. Philosopher Eva Jablonka, the biologist Marion Lamb, wrote in their 1995 book, Epigenetic Inheritance and Evolution, the Lamarckian Dimension. In recent years, molecular biology has shown that the genome is far more fluid and responsive to the environment than previously supposed. It has also shown that information can be transmitted to descendants in ways other than through the base sequence of DNA. We're back to where we started in this chapter, the environment. In my own work in the laboratory, I saw over and over the impact a changed environment had on the cells when I was studying. But it was only at the end of my research career at Stanford that the message fully sank in. I saw that endothelial cells, which are the blood vessel lining cells I was studying, changed their structure and function depending on their environment. When, for example, I added inflammatory chemicals to the tissue culture, the cells rapidly became the equivalent of macrophages, the scavengers of the immune system. What was so exciting for me was that the cells transformed when I destroyed their DNA with gamma rays. These endothelial cells were functionally enucleated, yet they completely changed their biological behavior in response to inflammatory agents, just as they had when their nuclei were intact. These cells were clearly showing some intelligent control in the absence of their genes. 20 years after my mentor, Irv Konigsberg's advice to first consider the environment when your cells are ailing, I finally got it. DNA does not control biology, and the nucleus itself is not the brain of the cell. Just like you and me, cells are shaped by where they live. In other words, it's the environment, stupid.